Welcome to Mr. Chalk's Revision Tips.
When answering exam questions, there are a few key things to remember, right? So just remember, exam technique is a key part in preparation for any exams, and it's something that you'll pick up the more you go and you do past papers. Right, so many questions in exams are repeats of what will be in old exams. So going and looking at lots of generic questions beforehand is a good idea. When going and answering and questions and reading them, there's a few things just take into account. Make sure you go read the question at least twice so you don't make any mistakes when you're reading them. As you're reading them, go highlight key information. Don't make assumptions about what the question is asking. Make sure after you've answered it, you go check your work for daft errors that you might make. And if it involves going and answering something where there's some calculation, make sure that you write your workings out down. Right, let's go and have a look at the question.
Sensory neurons detect a range of different stimuli. So a receptor converts stimuli into a nervous impulse, and that impulse is called a generator potential. So there are various different types of stimuli dependent on the different sources where it comes from. Right, however, all of this is referred to as sensory transduction. So this occurs when a stimulus is detected by this receptor, which generates a graded action potential or a generator potential, which is then moves down a neuron. If that is strong enough, the graded potential causes the sensory neuron to produce that action potential. This is then relayed to the central nervous system. So, Pacinian corpuscles are one of four major mechanoreceptors. Mechanoreceptors meaning that they respond to mechanical stimulus. So they have nerve endings in the skin and are responsible for detecting vibration and pressure. So a receptor that detects a mechanical pressure transduces it, so turns it, into another form of energy. We find them in the legs, hands, ligaments, places like that. So if you can see this diagram just here, it does look a little bit like an onion when it's cut open. So the Pacinian corpuscle is found in the center of various layers of tissues that separate or are separated by gel-like substances. So each of these layers are physically separated. So within the plasma membrane, there are protein channels, right? So the protein channels are there, which allow sodium ions to move through when open. So the sodium channel is also known as a stretch mediator because you need mechanical pressure to stretch them open. Sodium channels. In a normal state, the stretch-mediated sodium channels in the sensory neuron membrane are too narrow to allow sodium ions to pass through them. It's important that when we're answering these questions, we're always referring to the sodium as sodium ions. Don't just talk about sodium atoms. So the neuron of the Pacinian corpuscle has a resting potential. So when pressure is applied, the corpuscle changes shape. So this causes the membrane surrounding the neuron to go and be stretched open. So when the membrane stretches, the sodium ion channels widen. So sodium ion channels can now diffuse across the membrane, or they can move down that electrochemical gradient. So the influx of positive sodium ions changes the potential of the membrane so it becomes depolarized. Right? This results in a generator potential being produced. So we'll go and have a look at a question that kind of focuses around this. So the skin is important in enabling the body to detect changes in the environment. Okay, we know that. This allows the body to respond to these changes. Various types of sensory receptors are located in the skin. Right, all of that should just be kind of common sense. So figure 2.1 shows a photomicrograph of a transverse section of a pressure receptor, right, which we've just looked at. So that is that diagram, or that photo just there. So figure 2.2 is a diagram of a longitudinal section through a Pacinian corpuscle. So we're looking at this diagram just here. So the tip of the sensory receptor is not covered in a myelin sheath. Right, so we can see just there, so that's where it's talking about, that is not covered in a myelin sheath, whereas the rest, as we go and we move down the axon, is covered in a myelin sheath. It's also worth noting that these bits that are surrounding it are called lamella. The Pacinian corpuscle is a type of biological transducer. Remember what we said about transducer, it's turning one type of energy into another. So as pressure stimulus is exerted on the corpuscle, right, the lamella, which we've just gone and highlighted, is compressed and exert pressure on the tips of the sensory neuron. The plasma membrane of the tip of the neuron becomes deformed and more permeable to sodium ions. So this region of the neuron becomes depolarized, reaching a threshold potential, and an action potential is therefore generated. So we go and we look at some of the questions that lead on from that. So we're looking at why is the placental corpuscle described as a transducer. Right, remember the key things is it's converting one type of energy, so in this time mechanical energy, into another or different form of energy, so electro energy. Next, so deformation of the plasma membrane of the tip of the neuron causes the membrane to become more permeable to sodium ions. Suggest why, right? So the point is you're suggesting why. So remember, whenever we go in and we're answering these questions, it's all about getting in as much detail as possible. So you need to talk about the fact that the pressure is causing the sodium ion channels to go open. So that goes and leaves gaps or spaces that are large enough for the sodium ions to move through, so they can move through that phospholipid bilayer. And finally, so the generation of an action potential follows the all or nothing law. Right, explain what this means. Right, remember, your all or nothing law effectively is talking about a stimulus being strong enough to go and cause an action potential to be generated. So we can talk about the fact if the stimulus is not strong enough, then your threshold value is never reached. So therefore, depolarization of the membrane never happens or is insufficient, which means that an action potential will not be generated.
So hopefully you have found that useful. If you've got any questions, then drop it in the comments below, and we'll be uploading a lot more. Right, good luck, folks.